And hello and welcome to everyone attending in person and virtually. So I'm very excited to tell you all about the work I've done during my PhD. Oh, clickers. Okay. So every day we brush our teeth, which removes the gritty film that we can feel on the surface. This film is a biofilm, which if left unattended can form dental plaque. You're also probably very familiar with hearing about food recalls due to bacterial outbreaks, such as the one shown here, which are in part due to the formation of biofilms on food industry infrastructures and in water distribution pipes. Biofilms can also form on a variety of medical devices and in tissues. In fact, biofilms are so widespread that they are responsible for up to 80% of human infections and are therefore a major challenge in medicine. Pseudomonas aeruginosa is a gram-negative rod-shaped bacterium able to form a biofilm shown here in this scanning electron micrograph. P. aeruginosa is ubiquitous, able to thrive in diverse environments, including in the various medical devices and tissues marked with asterisks here. P. aeruginosa is a prevalent pathogen in a hospital environment, causing over 50% of hospital-acquired infections due to the transmissibility between devices and medical and tissues. As well, it has high antimicrobial recalcitrance. Now, this bacterial species is notorious for the chronic infections seen in the lungs of individuals with cystic fibrosis, CF, a genetic disorder. So what exactly is a biofilm? Biofilms are microbial communities encased in a self-produced matrix established to be a dominant microbial life form. The biofilm life cycle involves 4 general steps. The first stage is the formation of an initial aggregate of cells, either by adhesion to a surface or initial cohesion in solution. The second stage is the secretion of a matrix, represented by the green here, forming a microcolony. The third stage is growth and maturation and the emergence of an organized 3-dimensional architecture. Now, depending on the species and environmental conditions, 3 distinct types of biofilms can form at this stage. They can be surface-associated, submerged biofilms, and pellicles, which form at an air-liquid interface. The last stage is dispersion, where cell aggregates and/or planktonic cells are released passively or actively and begin a new cycle of biofilm formation. The self-produced matrix in biofilms contains chemically and functionally diverse extracellular polymeric substances, or EPS, such as proteins, extracellular DNA, and exopolysaccharides, which is the component studied in the Howell Lab. Now, in tandem, the EPS create a unique environment, allowing the biofilm community to have unique properties than non-biofilm forming bacteria. Exopolysaccharides are a crucial component of the biofilm matrix, as they are involved in surface adhesion, cell-to-cell interactions, forming a structural scaffold, and providing protection against antimicrobial and host immune responses. Exopolysaccharides produced by microorganisms are structurally diverse with distinct monosaccharide compositions. P. aeruginosa can synthesize 3 distinct exopolysaccharides. Shown here is the growth of P. aeruginosa colonies from the sputum of an individual with CF and a chronic lung infection. There are 2 colony types: mucoid colonies producing the exopolysaccharide alginate, and wrinkly colony variants, which are producing the PEL which are producing the PSL and PEL exopolysaccharides. So the focus of my work is on the PEL exopolysaccharide. PEL exists as a high molecular weight cell-associated and low molecular weight secreted form, but why or how these two forms exist have not been probed for in the literature. The PEL exopolysaccharide was recently found to be 50% deacetylated, rendering it cationic. Confocal imaging of mature PEL biofilms visualized using fluorescent staining showed where green represents biomass and red for PEL, showed that PEL biofilms form a characteristic mushroom shape and that PEL localizes to the biofilm stalk and periphery. Shown in yellow is DNA, which also localizes to the biofilm stalk. So other in vitro data shows that the PEL DNA interaction confers antibiotic and mucolytic tolerance. So how is the PEL exopolysaccharide made? In gram-negative bacteria, a prevalent mechanism to assemble and export exopolysaccharides outside of the cell is the synthase-dependent pathway. The polysaccharide component is represented by the gray hexagons here. There's a multi-protein synthase complex that consists of a membrane-embedded glycosyl-transferase and a copolymerase that binds the secondary messenger cyclic di-GMP for post-translational regulation. Transport of the exopolysaccharide across the periplasm is guided by export machinery and exported through a beta-barrel porin into the extracellular milieu. In the periplasm, modifying carbohydrate active enzymes can be found, which alter the physicochemical properties of the exopolysaccharide. The PEL exopolysaccharide is synthesized using this pathway, and shown here is our current PEL biosynthetic model. PelA is the modification enzyme in this system. PelA is bifunctional, so it has both a glycoside hydrolase and a deacetylase domain. These activities would modify the PEL polysaccharide, but to really get to what these modifications are doing, the complete structure of the PEL polysaccharide was not fully known, so we couldn't really address this. So we set out to determine this, where I optimized the protocol to isolate PEL and provided this to Francois Lemeuf in the Shepherd Lab for mass spectrometry and NMR analyses. GCMS analyses revealed that PEL contains N-acetylgalactosamine or GalNAc and galactosamine GalN that is 1,4 linked. MALDI-TOF and MSMS indicated that the monosaccharide arrangement of PEL contains predominantly GalNAc and GalN repeat units. And lastly, NMR analyses showed that PEL contains an alpha anomeric configuration. So we determined the complete structure of PEL to be a polysaccharide predominantly composed of a dimeric repeat of alpha-1,4 GalNAc and GalN. And the data is shown in our recent communications biology paper. Next, we wanted to focus on the role of each of these domains in the context of PEL biosynthesis. So focusing firstly on the deacetylase domain. So it's clear that the deacetylase domain deacetylates the PEL polysaccharide, and previous work has identified catalytic point mutants abrogating this activity in vitro on a pseudo substrate. In addition, it was also found that deacetylase activity is necessary for the formation of biofilms. Wrinkly colonies, indicative of exopolysaccharide, seen in the wild type, was lost in the PelA deacetylase mutants. They were instead smooth. Deacetylase mutants also formed no pellicle, and they had reduced adherence. So it's hypothesized that deacetylation, which would render Pel cationic, is necessary for Pel export into the extracellular milieu. PelC, anchored in the outer membrane, forms a funnel, and it's proposed to draw the Pel polysaccharide to its electronegative periplasmic face and guide it to the beta-baloporin to PelB for export. So to test this hypothesis and determine if Pel or fully acetylated Pel is exported, I probed the cell-associated and secreted fractions from an engineered Pel overproducing strain, PBAD Pel, and used WFL-HRP, a lectin that recognizes terminal GalNAc. And so this lectin would recognize fully acetylated Pel if it were exported. The wild type formed cell-associated and secreted Pel. The negative control strain, so deletion of the glycosyltransferase PelF and the entire PelA protein did not, as expected. The complementation strain looked comparable to wild type. And interestingly, the deacetylase mutant strain formed no cell-associated and secreted Pel. So to corroborate this dot plot, I also performed Congo red colony morphologies with these strains. The wild type formed a wrinkly colony, indicative of exopolysaccharide production, and the deacetylase mutant was smooth, further supporting that Pel is not found in the extracellular milieu. I also performed standing pellicle assays to look for the formation of a pellicle, and the deacetylase mutant formed a turbid culture, confirming it's unable to form a biofilm. So our finding that PelA deacetylase activity is necessary for the detection of Pel in the extracellular milieu, and thereby for Pel biofilm formation, makes it an attractive target to identify Pel biofilm inhibitors. So there are three proposed targets to inhibit biofilms. The first is inhibition of adhesins to prevent the initial adhesion stage of biofilm formation. The second is inhibition of the secondary messenger cyclic diGMP, which is involved in regulating EPS secretion machinery. And the third is inhibition of quorum sensing, a cell-cell communication mechanism that's involved in biofilm maturation. So targeting an exopolysaccharide modification enzyme has not been a proposed strategy to inhibit biofilms. So we sought out to determine if this could be a viable strategy by developing a high throughput screen targeting PelA deacetylase activity in vitro. The high throughput screen was performed by the Spark Biocenter and identified 233 hits. Subsequently, a reconfirmation screen identified 56 hits. So I took these 56 hits and I subjected them to a crystal violet assay, which served as a secondary assay to determine which compounds can inhibit biofilm formation. Pseudomonas cultures were grown with a hit compound overnight and then subsequently stained with crystal violet to quantify adherent biomass. I performed this assay using the PBAD Pel overexpression strain and omitted any compounds that had bactericidal activity, as these would be acting off target by killing the cells. We discovered 4 compounds able to inhibit PBAD Pel biofilms. One compound was found to inhibit Pel biofilms formed by multiple strains. In collaboration with the NITS lab, we confirmed the structure of this compound, identified its active form, and synthesized it in-house. This compound, which I'll refer to as 54O, robustly inhibits biofilms formed by the aeruginosa strain PA14, which intrinsically produces Pel biofilms, and has an IC50 of 1.8 micromolar. So to identify the essential elements in the structure of 54O, we performed structure-activity relationship studies by testing analogs of 54O for Pel biofilm inhibition. So we identified compounds that did not inhibit and did inhibit PA14 biofilms. So this informed us of key features for inhibition indicated here, and that 54O was the most robust Pel biofilm inhibitor. Another compound, 54PH, was comparable to 54O, and so moving forward, we worked towards determining the mode of inhibitory action of both 54O and 54PH. So first, I performed growth curves using PA14 to determine whether 54O and 54PH have antimicrobial activity. So at high concentrations, well above the IC50 value, there are bacteriostatic but not bactericidal effects. And that's indicated by the decreased exponential phase. So this supports that the inhibitory modality of 54O and 54PH are not by bacterial killing. In collaboration with the Melnick Lab, we next sought to examine whether exogenous 54O or 54PH treatment was cytotoxic to mammalian cells, specifically to IMR90 human lung fibroblast cells. So to examine if the cell compounds influenced cell morphology, we performed a cell rounding assay using Pseudomonas to obtain a cell rounding index, CRI, where a lower CRI is indicative of increased cell rounding, which precedes apoptosis. The Clostridium difficile toxin TCDB served as a positive control for cell rounding. So 54O was not significantly different for cell rounding at concentrations above and below the IC50 value compared to TCDB treated cells. So this means it does have a cytotoxic effect. 54PH, on the other hand, did not influence cell morphology to the same extent. It had a significantly higher CRI than TCDB treated cells at the highest concentration tested. With the Melnick Lab, we also examined the cytotoxicity of the compounds to human lung fibroblast cells by performing a cell titer blue cell viability assay to quantify metabolically active cells. So examination of the cellular viability of IMR90 cells treated with 54O revealed a concentration-dependent decrease in viability regardless of the media used. Now, in comparison, 54PH did not compromise cell viability, even at concentrations where PA14 biofilm adherence is inhibited. So this suggests that 54PH has a therapeutic window, which are a range of doses with a therapeutic response that does not cause an adverse effect in patients. So combined, these results reveal that 54PH is less cytotoxic than 54O. I then generated an IC50 curve using recombinantly expressed PelA and a fluorogenic pseudosubstrate to detect esterase activity in vitro. So with increasing concentrations of 54O, PelA's esterase activity decreased. So this titratable response supports that 54O is targeting PelA esterase activity. Interestingly, 54PH did not have a titratable response, revealing it is not targeting PelA and has off-target effects. So I then performed enzyme kinetics using the same assay to determine the mode of inhibitory action of 54O on PelA. Michaelis-Menten analyses revealed that 54O is a non-competitive inhibitor. So this means 54O has affinity for both free enzyme and enzyme substrate complex, and that the compound bounds to a site distinct from PelA's deacetylase active site. Future work involves further delineating the binding of 54O on PelA, which will be facilitated by the structure of PelA recently solved by Jamie in the Howell Lab. Also, to determine the target of 54PH. So the significance of this work is that it provides a unique proof of concept that targeting exopolysaccharide modification enzymes is a viable strategy to identify novel antibiofilm compounds for drug development. So next, we wanted to examine PelA's hydrolase activity. So recombinantly expressed PelA hydrolase, the recombinantly expressed domain rather, is able to disrupt Pel biofilms. So that indicates it's active. An E218A catalytic point mutant no longer has this activity. The structure of this domain and specificity is known, and it's established to play a role in biofilm dispersion. So what's unknown is what role this activity plays in the context of Pel biosynthesis. So to begin investigating the role of the PelA hydrolase, I performed a crystal violet assay, which quantifies biofilm formation via adherence. So wild-type PA14 formed a robust biofilm in contrast to our delta PelF and delta PelA negative control strains. A complementation strain rescued biofilm formation. And very interestingly, our PA14 hydrolase mutant strain had significant increased adherent biofilm biomass relative to the wild type and the complementation strains. Now, cell-to-surface adhesion is known to influence overall biofilm morphology by altering internal forces. So the decreased adhesion of wild type relative to the hydrolase mutant implies that there's an alteration in overall biofilm morphology. CFU counts, or colony-forming units, revealed that the strains had no compromise in cell viability, and transmission electron micrographs of the strains shown here showed normal rod-shaped cellular morphologies for Pseudomonas aeruginosa. So this means that the increased adherence by the hydrolase mutant strain is not due to growth defects. So why is this increased adherence happening? So to begin answering this, I took a deep dive into the literature and I came across the term matrixome, which describes the chemical and functional roles of EPS in biofilm assembly. So current biofilm literature is really pushing to view biofilms as more sophisticated entities to better reflect their complexity. Studies of the matrixome have revealed that EPS can be grouped into two major categories based on their location in the matrix. They can be secreted extracellularly and broadly contribute to biofilm structure integrity and protection from antibiotics and host immunity, or they can be associated with the cell surface and promote adhesion. Now, together, both forms of EPS contribute to the biomechanics of the biofilm. So what this means is that it contributes to how the biofilm reacts to forces and external stimuli. So knowing that the Pel exopolysaccharide exists in a secreted and cell-associated form, we hypothesized that the increased adherence we observed in the hydrolase mutant strain could reflect an alteration in the ratio of secreted and cell-associated Pel. So to probe for this, I performed a dot plot using the PBAD Pel strain background and the polyclonal Pel antibody. So taking you through this dot plot, we can see that the wild type strain produced both cell-associated and secreted Pel. As expected, delta PelF generated no Pel whatsoever. The delta PelA strain shows a faint signal in both fractions, and since this strain forms no biofilm, we hypothesize that this is contributable to background, perhaps due to the detection of sparse amounts of acetylated Pel released by cell lysis. The complementation strain, which forms a biofilm, produces cell-associated and secreted Pel. And remarkably, the hydrolase mutant strain produces only cell-associated Pel. So what this tells us is that a PelA hydrolase activity is responsible for generating low molecular weight secreted Pel. So we next wanted to examine if the absence of secreted Pel would influence biofilm architecture by growing flow cell biofilms and imaging using confocal microscopy to detect overall biomass, and Pel using WFL. So the upper panel here shows a top-down view of the biofilm, and the bottom panel here a side view. So the wild type formed a robust mushroom shape where Pel was detected in the biofilm stalk and periphery as previously established, and this is reflected on a WFL dot blot with cell-associated and secreted fractions. Delta F and delta A formed no biofilm or detectable Pel. The complementation strain looked comparable to wild type. And very interestingly, a hydrolase mutant strain had no detectable Pel using WFL for either confocal microscopy and dot blot analyses, even though it forms a mushroom-shaped biofilm. So what could be going on here? So the WFL lectin recognizes terminal GalNAc. So we hypothesized that in the wild type strain with an active PelA hydrolase, there would be varying lengths and therefore multiple individual polymers providing increased binding sites for WFL. The hydrolase mutant, on the other hand, with an inactive PelA hydrolase would generate longer and fewer individual polymers, providing decreased binding sites for WFL. So, though secreted Pel does not appear to be required for microcolony formation, it does appear to be modifying polymer length. Okay, so at this point, we have discovered that PelA hydrolase activity decreases adherence and is responsible for generating low molecular weight secreted Pel, implying it modifies polysaccharide length. So knowing that EPS components contribute to biofilm biomechanics, we wondered if secreted Pel could specifically have an influence. So we examined this by performing different biofilm assays. The first was to examine pellicle formation in glass tubes and their integrity by tipping the pellicles into a petri dish. So pellicles form to increase access to oxygen. And they're also proposed to float due to the biofilm matrix preventing mixing with the liquid media and by trapping of gas accumulated through metabolic processing. So PA14 formed a robust pellicle at the air-liquid interface, and when tipped, the pellicle maintained its physical integrity as a single piece structure. Delta F and delta A did not form pellicles, and the hydrolase mutant formed a pellicle with increased sagging relative to the wild type. Now, tipping the contents of the hydrolase mutant into a petri dish revealed that the pellicle had poor integrity. So this tells us that the wild type, which produces both cell-associated and secreted Pel, has increased pellicle cohesion and buoyancy relative to a hydrolase mutant. So what this implies is that secreted Pel could promote matrix interactions and oxygenation that prevent mixing with liquid media and the release of gas, promoting cohesion and buoyancy. So next, we wanted to examine colony biofilm morphologies as wrinkling occurs due to the production of exopolysaccharides. In addition, PA14 wrinkling is known to occur as a strategy to increase the surface area to promote access to oxygen and balance the intracellular redox state of biofilm cells. So robust wrinkling was observed in wild-type PA14, which has both central and spoke wrinkles. Delta F and delta A have smooth colony biofilm morphologies, which are expected for non-biofilm forming strains. Intriguingly, the PelA hydrolase mutant had decreased wrinkling. There are no spoke wrinkles. It has a thick central wrinkle. So this has really interesting implications for the forces driving wrinkle development. Shown here is a side view of the colony biofilms. So colony biofilm wrinkling is established to occur in 2 stages. The first is cell verticalization to reduce stress generated by cellular growth, and this creates the central wrinkle. The second stage is cellular ejection of the verticalized cells, creating the spoke wrinkles. So the hydrolase mutant having only a central wrinkle means that only verticalization is occurring. So this suggests that the generation of secreted Pel alters forces and is necessary for cellular ejection. Now, the interesting with this phenotype is that I noticed it looked similar to when BSLA is deleted. So BSLA is a secreted protein in Bacillus subtilis colony biofilms and is a major contributor to colony hydrophobicity and wettability. So it's known that colony biofilm wrinkling and surface chemistry can influence the wettability, or the ability of a liquid to spread over the biofilm surface, which influences the penetration of antimicrobial liquids. So we wanted to examine if wettability was altered by performing contact angle hydrophobicity experiments, where a drop of water is directly added onto the colony biofilm and the contact angle measured. So the Howell Lab measured the angles for us, and all the contact angles were less than 90 degrees. So this means the surfaces were all hydrophilic or wettable. However, all of the mutants had significantly lower contact angles than the wild type, so they were more hydrophilic. So this reveals that the wild type, relative to the hydrolase mutant strain, has decreased surface wettability. This implies that the generation of secreted Pel could aid in limiting the penetration of antimicrobial liquids. We then wanted to examine mechanical properties by pursuing rheology, the study of the flow and deformation of matter that's established to reflect biofilm composition and structure. So our collaborator, Dr. Aaron Gloak, performed indentation rheology, which involves measuring the force required to compress the colony biofilms. So she calculated the Young's modulus, which is a parameter that measures stiffness. So all the mutants had a significantly lower Young's modulus than the wild type. So what this tells us is that a PelA hydrolase mutant colony biofilm is soft and flexible, and that PelA hydrolase activity increases the stiffness of biofilms. So this increased stiffness of wild type relative to the hydrolase mutant implies that the generation of secreted Pel impacts diffusion, and this could influence antibiotic and host immune response tolerance. So knowing that the generation of secreted Pel influences biofilm biomechanics, we wondered if virulence would be affected as well. In collaboration with the Renke Lab, Alex performed an infection model using C. elegans, where death of the worms is due to infection in the worm gut through consumption of PA14. So relative to an avirulent control, wild type, and delta PelF, a non-biofilm forming strain, the hydrolase mutant had significantly increased killing kinetics. So there was only about 20% worm survival after 72 hours. So the hydrolase mutant kills a C. elegans host faster than both wild type and a non-biofilm forming strain. To corroborate these results in the C. elegans survival assays, in collaboration with the Howell Lab here at SickKids, Oksana and I examined host survival in an established Drosophila oral infection model that's been developed to study aeruginosa biofilm infections. So as a negative control, Drosophila were fed 5% sucrose. The hydrolase mutant had significantly increased rate of Drosophila killing relative to wild type. So in both C. elegans and Drosophila infection models, the hydrolase mutant strain killed the host faster than wild type, suggesting it's more virulent. So though the hydrolase mutant has compromised biomechanics, it has increased killing kinetics relative to wild type, indicating that secreted Pel reduces P. aeruginosa virulence. So this is significant because it provides the first evidence implicating the Pel exopolysaccharide in virulence modulation and suggests that secreted Pel is beneficial to the host. So how could secreted Pel be beneficial to the host? So we hypothesize that in a hydrolase mutant strain with no secreted Pel, there's an absence of ionic interactions. In the wild type where secreted Pel is present, we think it could sequester reactive oxygen species, or ROS, released by a host's immune response. The sequestration of ROS by secreted Pel could trap ROS and provide the host protection. Now, this ROS sequestration could also provide the pathogen protection by conferring host immune response evasion that exopolysaccharides are reported to provide. Additionally, secreted Pel could also be responsible for conferring the established Pel interaction with DNA that's responsible for conferring antibiotic tolerance, and it could interact with oxygen to allow for intracellular redox state balancing. So in collaboration with the Parkinson Lab, we have found that Pel biosynthetic loci are widespread across bacteria, found in over 500 gram-negative organisms and over 900 gram-positive organisms. So this means our findings have far-reaching implications. So future work could examine if secreted Pel is responsible for ionic interactions and if the phenotypes we observed are generalizable for other Pel-producing species and glycoside hydrolases involved in exopolysaccharide biosynthesis. Now, this work is significant because we have identified a novel role of glycoside hydrolase activity, which has advanced our understanding of Pel and the matrixome. So with our findings, we propose that secreted biofilm components be referred to as matrix associated to better reflect their influence. So in summary, Pseudomonas aeruginosa biofilms containing the exopolysaccharide Pel contain PelA, a bifunctional modification enzyme that modifies Pel. In the context of Pel biosynthesis, deacetylase activity is necessary to detect Pel in the extracellular milieu and thereby for biofilm formation, which we have demonstrated is a viable target to identify biofilm inhibitors. Hydrolase activity generates low molecular weight secreted Pel, which alters biofilm biomechanics and reduces virulence. So first and foremost, I would like to thank Lynn, my supervisor, for giving me the opportunity to pursue this PhD in her lab, which has helped me grow in many ways, and it's allowed me to meet many lovely people. I'd also like to thank my committee members, Roman and Mark, for encouraging me to do a PhD and for all of your helpful feedback over the years. I'd also like to thank all past Howell Lab members for all of your advice and support. I would especially like to thank Greg Whitfield for being a mentor and a role model for how I aspire to be both as a scientist and a kind human being. Thank you to current Howell Lab members, especially Andrea, for all of the emotional support you have given me. I've had the pleasure to collaborate with many lovely people, and I thank you all immensely for your hard work. Francois, thank you for your hard work on determining the composition of Pel. And thank you so much to Ben, who is so integral for the PelA inhibitor project. The story would not have been as exciting as it turned out without your chemistry expertise. I also thank Zach for his enzymology enthusiasm, and to Greg Wozni for teaching me the art of performing enzyme kinetics. I'd also like to thank Matt Parsick, Courtney, Julia, Joe, and Dan for all of your helpful feedback and advice over the years. I'd also like to thank all of my family, friends, and my therapist, Mila, for all of your love and support. encouragement these past 8 years. I also would really like to thank my cousin Amina Dami for being integral for the American Sign Language interpretation today, and of course to the interpreters Janet and Sarah for their hard work interpreting this seminar. Thank you for all of your attention, and I would be happy to take any questions in person or virtually.